Hi, everyone, and welcome to Night School. I'm Lynn.
And I'm Christina. Welcome.
And we are part of the Nightlife team at Cal Academy in San Francisco. For those of you who are new, Nightlife is our weekly Thursday night events where we mix science with cocktails, live music, and more. Night School is our at-home version where we bring you a new fun theme every week. So last week, we teased that we had some exciting nightlife news. And we're coming back next week. Nightlife and Night School will be next week. So we announced on Tuesday that Nightlife is coming back in the building on select Thursdays, starting next Thursday. I was about to say May 13th. So for those of you who are Nightlife, who have been to Nightlife before, come back and visit us. For those of you who are new, come check it out. This has been kind of a preview, so hopefully you'll be able to come. And we'll drop some links later to give you more information. But going back to tonight, So in March, we brought you all some extreme science, exploring extreme conditions that researchers and scientists go through to get questions answered. And tonight, we are actually going to bring you extreme life. So we'll introduce you to some incredible animals and organisms that are pretty extreme in their own right.
Like birds with very potent neurotoxin in their skin and feathers. So Dr. Jack Dumbacher returns. He's the Academy's curator of ornithology and mammalogy, and he's going to take us to the rainforests of New Guinea and talk about the questions surrounding these very poisonous pitohui birds, like where do they get the toxins and how do they use it? Also extreme, the mysterious desert oasis of Mexico's Cuatro Ciénegas Basin has super poor nutrient levels, a wildly fluctuating pH, sulfurous pool, and also microbes that have thrived there for 500 million years. So Dr. Valeria Souza is a microbiologist based in Mexico City who's going to tell us the story of this lost world and her work studying the evolution of microbial life. And finally, also very extreme, are wood frogs that freeze solid every winter, but thaw out for the spring, just in time for mating and everything else frogs have to do. Don Larson is an assistant professor at the University of Alaska Fairbanks, and he studies how they survive the Arctic weather, and we'll talk about these frogs and their incredible and unique adaptations. So great night ahead.
Yes. For those of you wondering in the comments, yes, we will still be doing night school weekly, at least until the end of June. So we'll be here with you. So don't worry. But as always, tonight's program is live. So say hi. Let us know where you're watching from. Same poll as always. Let us know if it's your first time joining or your night school regular. We will have Q&As after everyone's talk. So get your questions in the chat and we will turn it over to Jack.
Cool. Welcome, everybody. Thanks for joining. It's great to be here. Thanks for that introduction. I'm curator of birds and mammals at the California Academy of Sciences, and I get to go to a lot of cool places, but I think my favorite of all is New Guinea, and I do think it's one of the most extreme places I get to work. This is the island of New Guinea, just north of Australia, at the western edge of the Pacific Ocean, and it's just south of the equator, so it's all tropical. It's also the second largest island in the world, second behind Greenland. So it's the largest tropical island, larger than Borneo or Madagascar. And you can see it's still very green, and it still has mostly tropical rainforest across it. And it hasn't been destroyed as much as many of the other tropical rainforests in the world. And that's largely because it is so rugged that it's very, very difficult to build roads and maintain roads, and the amount of rain that they get with the steepness of the hills. And I think that's been one of the things that's really protected it. It's also made it very hard to do work there. And it's very extreme getting around because there are no roads, or very few roads that connect the villages. So to get around, when we're working along the coast, we often go in boats, from sailboats to other kinds of boats that'll get us along the coast. But once you move inland, when we're in the lowlands, sometimes we'll use the other waterways like rivers, and we'll use smaller boats like these canoes or motorized canoes. When we go farther inland into the mountains, then it's mostly airplanes. And this is Missionary Aviation Fellowship, or MAF. The missionaries use it to get to many of the smaller villages, but scientists use their airplanes too, because it's one of the only ways to get to many of these airstrips. And a lot of these are very short because the hills are so rugged that there's not a lot of flat space. And this one in particular, you can see it's kind of shaped like a giant ski ramp, and the plane sits at the top and zooms down the hill and launches itself over the valley, hopefully going fast enough to get some air. If that sounds scary, the really scary part is landing, because you're basically flying straight into a mountain and you get one shot at hitting that airstrip right. And then it's the trip uphill that actually slows you down. But we usually get there safely in some of these villages. And once we do get to these villages, it's really amazing because the local people live in a traditional lifestyle pretty much the same way that they have for a very long time. And they live with the land, and so they know a lot about the animals and the plants that are in the environment, and they are great to work with because of that, because of their deep knowledge of the land. Now, the planes only get you so far, and then the last bit you usually have to walk on foot, either for a few hours or a couple of days, depending upon where you're going. But it always pays off, because what you find in New Guinea is really, truly amazing. And because I study birds, I know the birds the best. But here's the world's smallest parrots. This is the yellow-capped pygmy parrot, and it's about the same size as our hummingbirds in North America, especially the hummingbirds of the Bay Area. So they're very tiny, about the size of my thumb. This is one of the largest birds in New Guinea. It's a little bit smaller than an ostrich, but it's still big enough to kill you if you get on its wrong side. And of course, some of the most beautiful birds in the world, like the birds of paradise. This is the magnificent bird of paradise. And many of these plumes and feathers are for their mating dances. And here on the left is the kingbird of paradise, and then the Rajiana bird of paradise. And on my first trip to New Guinea, I was there to study the Rajiana bird of paradise. And that was when I discovered this bird, the hooded pitohui, which is now my favorite bird in New Guinea. Because they're poisonous. And the first time that I had handled these birds, I was taking a couple of them out of the mist net to release them and let them go. And you can kind of see this little hook on the tip of the bill. And these birds are about the size of a jay, and they can easily scratch your hands with their claws and with their beak. And when they do that, I just licked my fingers, and my mouth began to tingle and burn and go numb. And I realized that there was something weird about these birds. And when I asked my local counterparts, they said, "Oh yeah, you shouldn't touch those birds. They're poisonous." And so that launched an entire research program for me. And over the years, we've been able to study these birds and their toxins and how they get them. And that's what I want to talk about today. So one of the first things that we did was we worked with researchers, chemists at the National Institutes of Health in order to isolate and identify the compounds that the pitohuis carry. And it turns out that they carry a steroidal alkaloid neurotoxin called batrachotoxin. It's a steroid because it has this steroid-like base. It's a alkaloid because it has this nitrogen-containing ring. And it's a neurotoxin because it binds to the voltage-gated sodium channels in our nerve and muscle membranes. And what happens is that it's the opening and closing of these channels that sends the electrical impulse down the nerve or muscle, which causes them to fire. And the voltage-gated sodium channels, when they're bound with batrachotoxin, open, but then they don't close. And this prevents the cell from reestablishing the ionic potential needed for the second firing. And it basically takes that nerve or that muscle offline. Because of this, and because it takes very few of these channels to depolarize the cell, they're among the most toxic natural substances known. And for me, when I first experienced this was the numbness and tingling sensation that I first got. But at higher doses, it can cause the nerves to totally shut down. It can cause paralysis. And eventually, if you get it internally, it can cause death because your heart can stop. It's also expected to affect a wide range of target species because the sodium channels are very conserved across all animal life. So basically anything with the central nervous system and with muscles that work like ours will be affected by these toxins. So, and these toxins, because they're called batrachotoxin, batracho meaning frog from Greek, and toxin, these were previously known from the Phyllobates poison dart frogs. Now, this is one of the Central American poison dart frogs. And now of all the so-called poison dart frogs, there are about 300 different species. Only 3 species were ever actually used by the Choco and Embera Indians of Colombia to poison dart tips. And this, Phyllobates terribilis, is the most toxic of those. And it has exactly the same toxins as the pitohui birds have. And in fact, just holding this frog in your hand can kill you. So once we knew what the toxin was, we wanted to understand a little bit about how the toxins were distributed within a bird, to understand where they're stored, and a couple of other interesting questions. And so this is a graph from some of that work. On the y-axis here, this is our measure of tissue toxin concentration. And on the x-axis, these are some of the different tissues that we tested. So first of all, let's look at muscle. So we found that muscle tissue has about 5 micrograms of batrachotoxin per gram of muscle. Now, just to put this into perspective, batrachotoxin is so poisonous that 0.05 grams of batrachotoxin will kill a mouse if injected within about a minute to 2 minutes. This means that every gram of muscle in a 65-gram bird has enough toxin to kill 100 mice. Okay, so even though this is much less toxic than some of the other tissues, it's still very toxic. And because muscle and heart and liver have those sodium channels, you would expect that birds would be poisoned by having the toxin in those tissues, but they're not poisoned. And so that's one of the questions we're going to talk about at the end of the talk. But let's talk a little bit about skin and feathers, because this is where the toxin concentration is the highest. And just to put this into perspective, if you pluck a single feather from the belly of a pitohui and pop it on your tongue, it'll cause your tongue to tingle and burn. And sometimes that sensation can last for several hours. And this means that these toxins being on the outside of a bird in the skin and feathers are something that an enemy would readily come into contact with. And so we think that these birds are probably using these toxins for chemical defense. And you notice that the birds are also this very stereotypical kind of orange and black coloration, the same as you see in monarch butterflies and other things that are toxic and chemically defended. So we want to begin thinking about chemical defense and ask whether toxins can protect pitohuis from their enemies. Well, the first thing you need to think about is, what are the enemies of pitohuis? And there are many. Here's an example of an external parasite or an ectoparasite of a pitohui. And these include things like mites and lice and fleas and ticks. And we've been able to do some very simple experiments to show that the toxin can act as an insect repellent to lice, but it can also act as an insecticide because it can shorten the lifespan of the lice. Okay, so it can potentially protect them from some of their ectoparasites, but we don't think that that's the whole story. There are also many snakes that can feed on a pitohui-sized bird, including the green tree python, this brown tree snake. And this is the snake that got into Guam accidentally and has decimated all of the vertebrate populations in Guam. And it's a snake that's endemic to New Guinea, something that pitohuis would have had to have evolved with. We've been able to show that both of these different snake species react very strongly to batrachotoxin and may be defended, the birds may be defended against them. We haven't done any experiments with goshawks and other large hawks, but there's at least 8 different species of large hawks that are big enough to take a pitohui-sized bird, and they should also have the sodium channels, and they should be sensitive to the toxin as well and potentially deterred. Some of our best evidence comes from human hunters. So in every village I've been to, the local people all hunt birds. That's a major source of protein for the local folks there. And the human hunters all know to avoid pitohuis. And so we know that the birds are protected at least from humans, and that the toxin has enough of a bad taste and a bad experience for humans that people won't hunt them. Another question that we had was, where do the toxins come from? Are the pitohuis producing it themselves, or do they get it from food? And there's several different reasons that we suspected that it was probably coming from their food, but being omnivorous birds in a very diverse rainforest, it's very difficult to nail down what the source may be. And so this story really comes from Avit. This is Avit here. This is his village of Harawana in the Eastern Highlands province. And I had the opportunity to work with him for a couple different years in that village. And he actually put together a study while I wasn't even in the village. And he worked with some of the elders in the village, and they all suspected this particular animal, the nanny sanny beetle. And nanny sanny it means poison beetle or bitter beetle, but it refers to the specific tingling, burning sensation. It's a lot like putting a 9-volt battery on your tongue. That you get when you taste the nanny sanny beetle. And they said it's exactly the same as you get from a pitohui. So that's their traditional knowledge and how they knew that it was this beetle. So Avit had collected a number of vials with these beetles in them and had the date and the locality and the collector's name on them. It was a beautiful collection. And when I went back to the village to see Avit, he gave me these and he said, go test these. I think this is where the toxin is. We took them back to the National Institutes of Health, shot those chemicals on our GC mass spec, and it was full of batrachotoxin. So we went back to some old stomach contents that we had collected of pitohuis, and we were able to show that this little beetle is eaten by the pitohuis, and that these nanny sanny beetles are so poisonous that the pitohuis would only have to eat a small number of them to have all of the toxins that they would need. And interestingly, if you hassle these beetles, they tend to turn over and curl up like this, and they exude little bits of little droplets from these glandular vesicles on their abdominal segments in prothorax and metathorax. And we think that this is probably where the toxins are stored in the beetles. So birds that might eat poisonous things and they accumulate these poisons in their body should be poisoned by the toxin. So one of the questions that we're investigating right now is, how do pitohuis avoid auto-intoxication? Why don't they get poisoned by their own poisons? And so there's 3 different major ways that animals avoid auto-intoxication. The first one is to just enhance the ability of detoxifying and eliminating the toxins. Whenever we have a drink of alcohol, that's what our liver and kidney does. The liver detoxifies the alcohol, the kidneys help eliminate it, and that's one way to deal with the toxins. Now, that's not going to help you if you're a pitohui, because you want to be able to accumulate the toxins and use them for defense. And because we know that the pitohuis accumulate them, we know that this is not the method that they use. Now, the second one is that there may be mutations in the target proteins that confer resistance. So, for example, this is the sodium channel in the membrane, and the toxin usually binds to the channel. Now, it's been shown in some other species that a couple of different point mutations in the sodium channel gene sequence can cause the channel to become resistant to batrachotoxin, and batrachotoxin can no longer bind to it. And so we sequenced the entire genome of the pitohui. We pulled out the gene sequences from all of the different sodium channels. There are actually 10 different ones expressed in different tissues and at different life stages. And then we tested several of these for their for whether or not they would bind batrachotoxin and whether or not they have any of these mutations. It didn't look like they had any of these mutations, but we were actually able to then clone these sequences into a bacterial vector and express them in the vector, and then actually study the pitohui sodium channel in those vectors and show quite conclusively that the pitohui sodium channels, every one of them, seems to be not resistant to the batrachotoxin, so they should be poisoned. And furthermore, we went back to the poisoned dart frogs to ask the same question, because there had been some papers in the literature suggesting that poisoned dart frogs are insensitive to the toxins, but we were able to show that actually the poisoned dart frogs are sensitive to the poisons and that they do not have any mutations in the target proteins. And so that's not the answer either. So this brings us to the last one, which is toxin sequestration. So and I should say, toxin sequestration is not just accumulating the toxins, but it's also being able to take the toxins and hold them in some way and usher them to places where they're not going to do any harm and keep them from doing any harm while they're on the way. And there is models for this. So there are some different sequestration systems that have been described in other animals. And there's some evidence in pitohuis that this may be the case. So for example, in the skins of pitohuis, we don't find that the toxin is just found willy-nilly in the skin, but these dark bodies, we have a stain here, and this is transmission electron microscopy, and these dark stains show that the toxin is accumulated in these multigranular bodies, and it's kept in the multigranular bodies where it can't do damage to other parts of the cell. And so we know that the pitohuis can sequester the toxins and that they do that in their skins, but exactly how they do that, we don't know. And this is what we're studying right now with our colleagues. And so this is the next thing that we want to study with the pitohuis. So in the future, we hope to get back to New Guinea so that we can continue to study how birds do this. And I think that there's going to be some great medical spin-offs from this work. And our colleagues at UCSF have been great collaborators in this research. And I'd like to thank everybody who's helped. And that's our little story about poisonous birds in New Guinea. And I'd like to thank all the people who've contributed to this work. Thank you.
Thanks, Jack. That was so interesting. We have a bunch of questions. The first one is the pitohui poisonous or venomous? What's the difference? And would you say there may be both?
They are poisonous, but not venomous. So venomous animals are animals that have an ability to inject their toxin and use it offensively. So if you think about a snake, it has fangs and it's venomous because it can inject that toxin into you from its fangs. Bees are venomous because they have a stinger and they can inject the toxin into you via the stinger. So the poison dart frogs are not venomous. They're poisonous because they contain the poison, but they basically use it for defense and they have no way to deliver it to something else. But if something tries to eat them, that thing will get a mouthful of poison and hopefully let them go. And that's how both the poison dart frogs and the birds work. That's a great question, though, because a lot of people don't understand the difference between being venomous and poisonous.
Let's see, what other questions? How potent is the toxin in claws or saliva, or is the toxin passed through just the claws or also saliva?
Well, so when we just handled the birds and then let them go, and you know, the scrapes on our fingers hurt, it stung, but it wasn't enough to, you know, get into our bloodstream and do any damage. But then if, you know, you lick your fingers, then it causes your mouth to tingle and burn. And, you know, we suspect that if you tried to eat a pitohui, you'd get a mouthful. If you were a hawk that had to pluck the feathers first, you'd get a mouthful of this poison, and it would be like hot chili peppers to them. It would, you know, it should deter them from wanting to eat anymore and not actually swallow them. Now, I know from colleagues who've worked in Central America that if you eat those poison frogs, or if a village dog, for example, eats the skin of a poison frog, it'll die. But we don't know, you know, most of the studies of toxicity have been done by injecting an animal with the Batrachia toxin. So we don't actually know what the LD50, the lethal dose if you ate them, would be. I have talked to one New Guinea villager who made the mistake of eating a poisonous bird. And he said it was a terrible experience. He does not wish to repeat it, but it didn't kill him. So.
Yeah, I wouldn't imagine wanting to go through that twice. Unlike the pitohuis, do the beetles and poison dart frogs produce their own toxins?
That's a good question. From all the evidence that we have, the poison dart frogs do not produce their own poisons. So if you bring them into captivity, they slowly lose their poisons with time. If you raise them in captivity, they don't produce any toxins. But if you take a non-toxic domesticated poison dart frog and feed them toxins, they readily accumulate the poisons in their skin. So all the evidence we have suggests that the poison dart frogs get it from some dietary source. However, no one's ever been able to figure out what the dietary source is for the poison dart frogs. They've never found it in anything else in Central or South America. So that's one thing that was very exciting about the pitohui work, is that we did find a potential source, but we don't know where the beetles get the toxin. The beetles are only about the size of a grain of rice, and they mostly eat smaller insects. Now, there was a paper that came out a few years ago suggesting that aurabatid mites actually produce a huge number of different chemicals, and some of the same chemicals that appear in poison dart frogs. And although we haven't found Batrachia toxins in the aurabatid mites, no one's really looked in New Guinea for them either. So it's possible that in fact, the beetles don't have the mavonolate pathway for making steroids. So the beetles, if they do produce the toxin, would have to at least get the steroids from something else. But we think that the beetles are probably getting it from some other source. So yeah, the mystery continues. We really don't know what the original source of Batrachia toxin is in New Guinea or in Central America.
Endless amounts of questions. Going back to what we just talked about like the saliva, would cooking the birds diminish the poison's effects?
Probably not, but there may be other things in your dinner that might react with the Batrachia toxin to affect its toxicity. But you know, if you think of other steroid-like chemicals, they are pretty resistant to cooking. And in fact, some of the techniques that we use for preparing the samples involve a little bit of heating. So they seem to be pretty stable compounds. And in fact, my colleagues at the National Institutes of Health have done some studies looking at some of the dart tips that were in Smithsonian's collection. And some of those dart tips were up to 100 years old, and they still were toxic enough to kill prey with. So there was enough toxin still in those dart tips. So it's a pretty stable compound.
Last question. Do you think there are any other poisonous birds that acquire toxins this way?
There are. In fact, the first paper that we ever published was the cover of Science, which was really great. But the downside of that is that everybody wrote in examples of poisonous birds that they knew about. And we actually compiled all those together and did a lot of research on it and published a really neat little paper. And I think it was, I think it was 1996. All these papers are available on my website, jackdumbacher.com. And it outlines some really interesting examples of poisonous birds. So for example, the native parrot to North America, the Carolina parakeet, was believed to be poisonous. And our great fathers of North American ornithology, like Alexander Wilson and John James Audubon, both did some experiments to test whether those birds were poisonous. And Audubon probably killed his dog, feeding them poisonous parakeets. And there's a couple of other really cool examples. The oldest example of a poisonous bird is actually from the Bible, but people still get poisoned by the European quail on certain flyways and during certain times of year in the Mediterranean, northern Algeria, parts of southern France, and probably because they eat something on the flyway. And so there's a couple of other really fun examples that I like to tell stories about. We probably don't have time to go into all of those, but yes, there are a couple of other examples of poisonous birds. Pitohuis are not the only ones.
Cool. Thank you so much, Jack. If you're interested in learning more, there's a link to Jack's website in the chat, and you can look up all the other poisonous birds. Thanks so much, Jack.
Thanks, everybody.
Up next, we have Valeria Souza.
Okay, hello. I'm from Mexico, and my work site is as different as it can be from New Guinea. It's in the desert. We don't have a jungle, and it's the place with less primary productivity, probably. And despite that, it's probably even more diverse than New Guinea, not for birds, but for bacteria. So I want to tell you about the miracles we can have in society if we become aware of nature. So Cuatro Ciénegas has been an amazing adventure. I cannot see my show. Okay. So it's the place with more stromatolites that we know and more diverse stromatolites. These stromatolites were the reef of the past. They are in the fossil record for 3 billion years. And they were the dominant communities for a very, very long time, till the Ediacardian life explosion before the Cambrian. And so in Cuatro Ciénegas, they have many shapes and forms. And when they calcify, they can form these reefs. However, they can be as humble as this microbial mat that is between my fingers. And microbial mat is what makes stromatolites, that are basically rocks with stripes. But these stripes are given by these layers, colorful layers, that contain all the biochemical cycles of life. So here, contained in between my fingers, are all the metabolic capabilities of the planet and the history, the metabolic history of life on Earth. First, down here are the black ones. They are the metanogens, is the first metabolism. That is an autochemiolithotroph. That means they make food out of rocks. After that is all the sulfur metabolism that is really very diverse. And let's remember that early Earth was basically a furnace of sulfur gases and magmatic fire. And then is the miracle of light. Here we have the two first photosyntheses, the purple and the green sulfur and non-sulfur bacteria. The purple develop and evolve the photosystem 1 and the green photosystem 2. And each one used different antennas to pick up low light. However, probably by recombination, these two photosystems end up in a new creature called cyanobacteria. And the lineage of that first cyanobacteria evolve in a way where two photosystems are together in a membrane and have very powerful antennas as chlorophyll 1 and 2, A and B. And these chlorophylls capture light of the sun, and as an accident, it breaks the water around them, releasing oxygen. And so bubble by bubble, these cyanobacteria transform Earth forever. On top of them is the diatoms, the first algae. And then we have a little twig and my fingers as a representative of the animals. And in between, there's lots of fungi that live inside the microbial mat. So we have all the story of evolution there, more or less. And how we know that this is the lost world? How we know that this is not just a place that looks like the past, but is the past? Because when we do molecular clock of 16S gene, that is a very conserved gene, in a very common bacteria called Bacillus, from a collection of Cuatro Ciénegas, we found that there's this branch that is only found in Cuatro Ciénegas and is very old as when Rodinia opened 800 million years ago and allowed the stromatolites to have bigger shores and capture the CO2 of the atmosphere into the reefs. And that precipitates what is known as the Cryogenian. The Cryogenian is about of two big glaciations that froze Earth for several million years. And the ice then broke the rocks, and the rocks had stored into them the most precious element for life, and that's phosphorus. Before the Cryogenian, phosphorus was very low in all the oceans, and life was therefore restricted because phosphorus is energy. And if you don't have phosphorus, you don't have ATP, and also you don't have DNA. So life was limited by phosphorus for most of its life, for 3 billion years, till the stromatolites capture the CO2 and precipitate a global glaciation. And then when the phosphorus came into the ocean, along with water and oxygen, the algae that had already evolved on top of the microbial mats were happy as hell and grew and turned the ocean green and captured then into their bodies the CO2 again, precipitating a second glaciation called the Marinoan. And again, the ice broke the rocks, and again, phosphorus was liberated, and then it was turned off the animals. And the first animals start to eat in those stromatolites and start to make burrows on top of the stromatolites. And those burrows start to release the nutrients that were stored on the marine sediment for billions of years. And then the algae was again happy because it was limited by nitrogen. And they got to eat in the water column and transforming an orange ocean into a blue ocean. And then Bacillus, this aerobic bacteria, was the first witness of a blue ocean and oxygenated marine sediment. So it's amazing that they're still here. And they're still here because this mountain that looks like a Star Wars starship, is the storage, the treasure cave where all these marine sediment was stored into their clay caves. And below this mountain, there's fire. There's magmatic fire, and that magmatic fire is feeding the water up, like in Yellowstone, but this is deeper. Probably is an anomaly in the earth crust that is feeding Cuatro Ciénegas, because here in Cuatro Ciénegas, two supercontinents open, Rodinia and Pangaea. So it is a very special place. And this magmatic fire is making the water goes up and down, and along its path, the water moves the microbes that are in very old sediments in the depths of the mountain. And that's the cause of stromatolites. To have stromatolites, you need magmatic fire, sun, and water. And no algae. They hate algae. Algae grow too fast. So this is the hypothesis. It rains, the water goes down through all the different layers with the different histories of life on Earth, and then the magma makes the water goes up and makes a pond with very diverse microbes, because there's the seed bank of these bacteria is enormous. Here we have a place called Churince that no longer has water because humans are overexploiting this aquifer by growing alfalfa in the desert, which is a very silly thing to do. And we have a place called Domos del Arciano, who has the most ancient bacteria more ancient than when Rodinia broke. They are anaerobic and they eat fire. They are archaea that eat fire, and some of them are even, they have been found in deep sediments, marine sediments, and they are here in Cuatro Ciénegas. So what's the problem with the water in Cuatro Ciénegas? The problem is here. You can see it in Google Earth. This is alfalfa fields. Each one of these dots is 70 hectares, and they're fed by central wells. And also these canals that were built in the 70s. And one of these canals, the Saca Salada, goes 80 kilometers away of Cuatro Ciénegas, exporting every day 90% of the water of the valley. And the purpose of these canals were to dry up the wetland in order to make it available for agriculture, which was a very dumb thing to do because it doesn't rain in Cuatro Ciénegas. So this is my Churince, the most studied site in Mexico by far. We studied it for 16 years, 100 scientists, as fast as possible, as deep as possible, in order to understand it before it dies, so it didn't die in vain. There's a collection of books in Springer about this site. And now this lagoon has herbs and trees on top of the stromatolites, and it is very, very sad. So we need to change. We need to transform this extremely precious and unique place in a place for conservation, in a place where the children have a future, not a dry, abandoned village, but a wealthy place where everybody is okay. And in order to do that, we transform the consciousness of the kids through art. And one of our previous enemies, that is Lala, end up being now our ally in conservation, because children are important, and we end up convincing the CEO of Lala. It was his daughter that made the convincing more important. And then we made a molecular biology lab, also in Cuatro Ciénegas, and the teenagers in the high school are a very important part of the genome discovery in Cuatro Ciénegas. They are the example of Mexico of the Nagoya Treaty, where they are going to develop new antibiotics, cancer research, and a lot of genetic resources for agriculture. And they're doing it in Cuatro Ciénegas, and the revenues of those discoveries are going to return to Cuatro Ciénegas for sustainable growth and education. And these same kids are changing the way the parents and grandparents are viewing the soil and the water, and how to restore the fertility of the soil after being totally depleted of nutrients and water by the alfalfa fields. And so these kids have the wisdom now, and they are the example of their society. And these same kids decide to close the canal, this canal that takes out 90% of the water, in October of last year. And we were so happy of this miracle, because this river that's running here was dry for 100 years when they start to make the canals, and now is reborn. And the stromatolites that were dry for 100 years, now they are tickling with life. And the turtles and the fishes and everything that is endemic and loves water is again happy with all this water. And the wetland is now 40 hectares bigger in just 6 months. And more miracles are expected, because this guy that was my student in 2004 now has a PhD in biotechnology, and he's the leader of an effort called Genesis, where he is an expert in biotechnology. He discovered several new antibiotics and many proteins that slow down cancer growth, and also several proteins that make the plants grow better, and especially all kinds of ways of growing up without phosphorus. The bacteria in Cuatro Ciénegas has been depleted of phosphorus for so long that have evolved all the strategies, all the possible strategies to live without phosphorus. And phosphorus is a very, very important resource for agriculture. And we are very close to being without phosphorus in just few years because the mines of apatite, those rocks that were scratched by the ice in the Cryogenian 700 million years ago, are now depleted by our work. So let's hope that those miracles happen. Thank you. I don't know if I was too fast.
I don't think so. Thank you so much, Valeria. That was great. So you mentioned, I love how the way you said at the beginning, like that you were holding all the metabolic history of life on Earth. How many other places in the world can you do that?
Well, microbial mats are in many places. Mm-hmm. Here is the story of life on Earth. Yeah. However, the microbes that are in this particular microbial mat are unique. They are very, very ancient. And that's the difference between these microbial mats and other places like Yellowstone or Guerrero Negro or Shark Bay, and even many lagoons in Mexico that they have microbial mats. But there are microbial mats in any other place of the world are new. Ones are ancient because the mountains store the story. The mountains store the microbes and the minerals of the early ocean. And they still here. And that's not really a surprise because a year ago, they just published a paper about microbes from the deep South Pacific that became alive, and they were 100 million years old. And they just needed sugar and some oxygen to be happy. And so life is extremely, extremely sturdy.
Yeah. And so you mentioned how ancient these bacteria are. About how old are they? And are they the oldest living organisms still on Earth?
I think so. Well, they have evolved. There's no way to stop evolution.
Right.
Evolution is a tic-tac that goes every day. However, they have been isolated in the marine sediments of these mountains for at least 1 billion years. But we have to study the genomes of the anaerobic bacteria. And we are doing that. And it takes a lot of money and lots of time because anaerobic bacteria and the metanogen archaea are very, very slow to grow. They take years to make a colony. And so we have to be patient and have the money ready when they grow. But I'm sure that they are at least 2 billion years old.
Wow. Yeah. Incredible. It's hard to wrap your mind around it.
Yes.
Yeah.
And they survive here. And so I think the big lesson of planet Earth is not the origin of life. Life started very early, once that water was liquid. Is the sturdiness of life, the survival skills. And these microbes survive because they help each other. Mutualism is much more important than fighting. And that's a lesson we should learn.
Yes, absolutely.
They survive together.
Yes. And then it's wonderful to hear the miracles that are happening. What's the future of conservation, and what are some of the challenges that you still have to face with protecting this place?
Well, this is a fragile miracle that has to be held by the people of Cuatro Ciénegas, and that's the role of the kids. The kids are really pushing society, and there's a social revolution happening because in these poor farmlands, the kids hold the promise of a better future. They are going to college and they are studying, and they have a lot of skills that the parents didn't have and didn't have those opportunities. And we want to equip their school with better labs, to have a soil lab in order to really restore the fertility of the land, and so they don't open more fields. They do good agriculture, not exploitive agriculture.
Right.
They change the cultures for more water-saving cultures. And we have to change economy. And I think biotechnology for the people is the future of Cuatro Ciénegas. But they need time. They need a little bit of time in order to develop those biotechnology promises. And for those to occur, they need the water in the ponds.
Yeah, yeah.
We need the wetland.
Here's a question. So many extremophiles have adaptations for one extreme that allows them to survive other types of extremities. So how have the bacteria and these stromatolites been able to survive like hundreds and maybe more years of dryness without water? How can they survive?
Well, they survive inside the mountain in little caves of clay. And clay, when it is in the form of little caves, store a lot of water. So it's a spongy world under that mountain. It's not dry, it's hot. And so they got from extreme hot to acidic or alkaline environment, as you can see from my fingertips, very salty environment. But they have, what we see is that diversity is the protector. So these communities are so diverse that they have resilience through many copies of their genes. So each one of these communities has zillions of different proteins in order to respond to each kind of different hazards, environmental hazards, and they do it together. It's by being together that they can survive everything for billions of years. And I think that's amazing.
Yeah, yeah. So you've been studying this area for 16 years.
21.
21 years.
Yes.
What's next?
With a student, 16 years.
Okay. What's next? Like, what's the next question you want to answer?
I want to demonstrate my hypothesis of the mountain as a storage. And I want to demonstrate the age. So I told you very out of my hand 2 million years, but I have to show that with molecular clock and not in a small sample, but in a big sample. So to explore these microbial mats will take my lifetime, no doubt about that, because each centimeter that we study is different from the next centimeter.
Wow.
So the diversity is so high that is mind-blowing.
Yeah. Yeah. That's amazing.
Yeah. And I think the answer is time. They have been for so long under that mountain going up and down with the magmatic heat that they have stored a lot of diversity and lots of microbial radiations inside that mountain. And that's what we find.
Well, thank you so much for joining us tonight. It was wonderful to have you. You can now take your dogs out of dog detention. Yeah, thanks so much. And also, I just wanted to say that the Academy's online magazine, Biographic, wrote a story featuring Valeria's work. I don't know, a couple years ago?
Yes, 2 years ago.
2 years ago.
Now, that story showed that Cuatro Ciénegas was dying, and now we rescue it. You need a second story.
Yeah, there needs to be an update. I'll let them know. We'll drop a link to that in the comments. But thanks again for coming. And up next, we have Dr. Dawn Larson.
Thank you.
Hi. My name's Don Larson. I am an assistant professor up at the University of Alaska Fairbanks. It seems like we've done a good job of having people from all over the world talk today. So I am up here in Fairbanks. We still have winter, and I'm going to talk to you today about wood frogs and how they overwinter. And my talk is entitled Sweet Treats. And you'll find out later on why we call wood frogs sweet treats. So you probably know something already about Fairbanks. The big thing is Fairbanks is very cold and the Arctic is very cold. We usually see temperatures down to -40 Celsius or colder. And the first fun fact I'm going to teach you is -40 Celsius is about the same as -40 Fahrenheit. So it's just cold at that point. And if I was to ask people in this talk about how animals survive winter, most of you and most of your children could tell me, or kids that you know could tell me something about how animals overwinter. They would tell me that bears hibernate. Well, there's some animals like birds that migrate and fly away. And then there's other animals, the big animals like moose or wolves, that stay active every winter. And this is most of what people understand about how animals survive winter. You can tell me about the cute things, the megafauna, the things that people like to have stuffed animals of. But I ask questions about how other things overwinter. I ask questions of, we have these wonderful endotherms, these warm-blooded animals like our moose, like our birds, but then we have some of the other cute things like our ground squirrel. These ground squirrels actually overwinter with an internal body temperature below freezing at -2 degrees Celsius. But I ask questions of where ectotherms or cold-blooded animals go during winter. What happens to them? I already mentioned that Fairbanks winters are quite cold. What happens to these animals that are cold-blooded that can't maintain an internal body temperature above environmental or ambient conditions? So that moose, it can go cold. We don't know the temperature a moose will shiver at. It's quite happy at -40 degrees Celsius or -40 Fahrenheit, or something like a muskox is quite happy. Those migratory birds have are quite well adapted to traveling long distances. Even our hibernators, like our ground squirrels or bears, are very, very happy in those conditions. They winter isn't a challenge for them. Being cold and hibernating isn't a problem. But when we start to talk about some of these other animals, it becomes a little bit different of a problem. So when you think of Fairbanks, Alaska, one thing you think of is, again, that cold, but there are places that stay unfrozen. So if you're a dragonfly nymph, which we have right here, they're going to overwinter in the water, where as long as there's enough ice, they can stay unfrozen that whole time. But other things like yellow jacket wasps, or some of our other insects like stink bugs or beetles, have a slightly different adaptation. And we're going to see that our little wood frog, and this is a young of the year from a couple years back, aren't going to be able to survive the same way. So one of the unique things about water is, although we think of the freezing point of water at about 32 degrees Fahrenheit, 0 degrees Celsius, water can maintain a liquid well below freezing. In fact, you can hold water, if you have small enough amounts of water, all the way down to -42 degrees Celsius before it spontaneously freezes. So for some of these smaller insects like yellow jackets, they actually use this adaptation, and they do what's called supercooling. It's this really cool adaptation where that water remains a liquid unless it's agitated. And what you see here is supercooled water. And it's not until something causes ice to build that this animal then begins to freeze. So for that yellow jacket wasp, if it can spend winter underneath a leaf in a place where it's not touching any ice, it's going to be very fine and it's going to survive great. But this adaptation only works for extremely small animals. You have to be tiny for this to happen because the large animals aren't able to maintain that low of body water that needs to happen for this. And this is actually something you can do at home if you ever want. You can take especially distilled water, the more pure it is, the better, and at very small amounts, and stick it in your freezer for a few hours and check the temperature. And then if you add a little bit of snow or dirt, or if you shake it, you can cause that ice to spontaneously freeze and see that nucleation happening. It's quite fun time, especially for kids. Although it's something I've done with everybody from the 55 plus crowd when I teach what's called OLLI, lifelong learning courses, all the way down to third graders and second graders when I've done things with them as well. But like I said, wood frogs are a little bit too big for that. So they can't survive winter. They can't survive those -40, -50 days by just maintaining unfrozen water. They are going to need a different adaptation. So our yellow jacket could spend winter unfrozen, but without any trouble. Our dragonfly nymph can find some place underwater, but wood frogs don't have that ability. For a couple of reasons. One, it takes to be able to survive underwater as long as wood frogs need. They need to both be able to slow their metabolism and have access to enough oxygen. And our ponds and lakes just are too anoxic during the winter for that ever to be an adaptation. So instead, what they do is we have our little wood frog, and they'll hop around until things start to get cold in September here. And they will make themselves a little, what's called a hibernacula hole. And so here's our wood frog. The one giveaway of something being a wood frog is a mask. Wood frogs have this white striped mask on their face. And so if you ever pick up a frog and you see that mask, that means it's a wood frog. And so during early fall for us, September, they begin to search for a place to overwinter, a hibernacula. And what they do is they dig a little hole. And this hole's only, a few centimeters deep or a couple inches in depth. And they pat down the ground and slowly encircle it to create this little hole in the duff. This is, again, we're in the Arctic, so our soil isn't exactly all soil. It takes a while for things to decompose. So the forest layer here is instead a lot of dead leaves that aren't all the way to that soil stage yet. So they bury underneath those leaves and wait. And here we have actually an uncovered frog in the spring, ice surrounding it, even ice on it. But this animal is very much alive and very, very happy. Because instead of supercooling like those yellow jacket wasps do, or staying warm enough to not freeze, this animal is going to freeze. It's going to freeze 70% of its body water and survive winter in this ice-encased state. In fact, here's a video of a frozen frog, and I want to point out this eye right here. If you notice down in here, there's all this white. That's ice. There is ice in the humor of this eye of this frog. This frog, and what we're going to see is this frog thawing out and coming back to life. So in this frozen state, this frog has no heartbeat. It has no brain activity. It can't move its muscles. Individual cells and tissues are surrounded and encased in ice. This animal isn't a functioning organism at this point. It isn't capable of all those things organisms need to do. It's individual cells, individual tissues separated off from each other. But every spring, and it's actually happening right about now, once the snow's gone, wood frogs are going to start coming out, and they're going to wake up. That ice will begin to melt, and their limbs and muscles and heart will all start to be able to move again, and they will start functioning as an animal again. And then, of course, since they are wood frogs, the first thing they do when they wake up is they get into ponds and they breed. So within this frozen state, let's play this again. We have 70% body water frozen, and we have ice surrounding cells. Any internal ice formation within a cell is lethal. So if wood frogs can't survive with their cells frozen, instead they survive with their extracellular water frozen. All the water outside of the cells is frozen. And we're going to talk about how they can do this, and this relates back to them being sweet treats, and how that plays into this. But as I said, no heartbeat during that period. They are just completely not alive as a whole organism. They are alive in the, like if you were to put a body part on ice, and it's only once spring hits that they actually come back to life all the way and are ready to be that wood frog again. So what does a winter look like for these wood frogs? And this is where things start to get interesting. Here we have temperature and time, date. So our winters, temperatures get below freezing in October of this year. Depends on the year, but usually October is when we see our first days that are going to stay below freezing. Here's our freezing point here, this line. And this stays this way until April and May. We actually have a very late winter this year, since we had -20 Fahrenheit in mid-April and got one of our biggest 3-day snowstorms ever. But wood frogs underneath that snow cover actually get some insulation. So here's our air temperature and a very typical Fairbanks winter of getting down to -40 in January, sometimes in February, but that's usually when it happens. But wood frogs are protected, and so are every other animal that lives under the snow, because that snow provides insulation. It's like a big down jacket, nice and puffy, and it really holds air well. So for them, a wood frog only gets down to -18 degrees Celsius for this specific one. We've seen temperatures down to -20 Celsius, and to give you an idea, -20 is about -5 degrees Fahrenheit. And what you see here is the days that they stay frozen in our hibernacula is right at the end of October, and they don't wake up until April and May, with some of our frogs from this year not getting out until May. So this is 200 days frozen, or about 7 months. And we've overwintered frogs over 2 different years doing this, and the really remarkable thing is we have what I call both wonderful and terrible data, in that we have 100% survival. It's wonderful because it shows that winter's not a bottleneck for these animals. So right now, if you asked me how long can a wood frog survive frozen, I can't tell you. If you ask me how cold they can go, I can't tell you. I don't have enough data because none died. If I had a couple frogs die, I could say, well, it looks like 7 months is kind of near their tip. But we don't see that. We see 100% survival. And we see these frogs just doing great down to -20 degrees Celsius. We know there's species of Siberian salamander that can go down to -35 or -40. So we would expect that wood frogs in Alaska could survive probably to those conditions, as it's possible to get a -40 day in November. The length of time, that's completely unknown. We don't know. We know that wood frogs go as far north as the Arctic Ocean, and a winter in further north than Fairbanks, even just a couple hundred miles north, is about a couple weeks longer. So it might be 8 months or so that they could survive frozen, but even 7 months. That means half their life is spent in ice, not really doing much but being frozen. That is remarkable. And I want to illustrate how remarkable that is. If you put a pint of ice cream or maybe a steak or some frozen veggies in your freezer, 7 months without freezer burn is pretty miraculous. These animals are not only just like not getting that freezer burn, not getting wrecked like we see our foodstuffs, they are coming out and breeding right away. They're doing a lot more than simply not going bad. We're seeing this just remarkable survival in these animals. How do they do it? Would be the next big question. This is, 7 months frozen. We can't do anything we're near that. And they do it by accessing glycogen stored in their liver. So glycogen are these long chains of glucose or sugar that are stored in their liver. And what they do is once they begin to freeze, and only once they begin to freeze, they start to process that glycogen into glucose, into sugar. They have a couple other cryoprotectants as well. They have a antifreeze glycolipid that's most likely embedded in the cells to prevent the cells from freezing. And they also produce urea, a precursor to urine, but that's not made in as high of levels as glucose. But think of that. bears get fat before winter. Birds plump up before they migrate. Arctic foxes change their coats in preparation for winter. Our wood frog decides that they are only going to start making the thing that's going to protect them, glucose, in reaction, not preparation for winter. So if there's no ice, if the winter's nice and warm, they won't freeze. But the moment ice touches them, they will begin this reaction of producing glucose, just massive and massive and massive amounts of glucose, and then pushing it throughout the body as quickly as possible. Since they only make glucose when they freeze, they're going to have a limited amount of time to actually make enough glucose. They're only going to have a few weeks of when that first ice starts to form, and when the temperatures actually stay below freezing for the whole winter. So they'll need to balance those two things out. And what's even more remarkable is how much glucose this is. So here we have glucose as a concentration, micro nanomoles per gram of fresh weight. And then we have a few tissues. So this is in the lab before they're frozen. This is in the lab when our traditional method of freezing frogs in the lab is always very slow. Give them as much time as possible, because they only make glucose in response to ice formation. So the idea is give them as much time while you're slowly freezing them. But then here's the field. This amount of glucose is enough to taste sweet. And not like, just semi-sweet. I mean, like as much sugar as a concentration as a soda. You could pop a frog liver, a frozen wood frog liver into your morning coffee. And they will be you will notice a nice sugary drink. These frogs have enough glucose, enough sugar floating through their tiny little 5 to 10 gram bodies to kill an adult human multiple times over. They have more sugar in their little body than I have in my blood right now. They have just massive and massive and massive amounts of sugar, all flowing through them, all so they can survive winter. And this is where things get a little interesting. So when we think of how cells freeze, the typical idea is I have my little cell here, and then we have ice crystals. But we think of ice crystals as kind of acting like a press and crushing our cell. So we end up with a burst cell that was crushed by that ice forming. Or the other thing that people think of is ice forms inside this cell. These are my little ice crystals. I apologize that I'm a terrible artist. And that ice causes that cell to burst. And so what a lot of people believe, and this is a very common preconception to have, is that ice and that glucose prevents that ice from forming in the cell and bursting the cell. And that's true to an extent, but not the full case. The other part of this is initially when this frog freezes and when this water forms, is we don't have that solid ice cube state. We have a slushy state. Of both ice and water, and surrounding this frog are solutes and water. And as ice begins to form, we start to because we're forming ice, we start to remove this water. And you're familiar with this if you've ever had like a freezy pop or made ice cubes at home. If you make ice cubes at home, the water is cloudy in some places and clear in others. And this is because as ice forms, it pushes solutes out of that ice matrix. The same reason why if you're sucking on a otter pop or a freezy pop, all the flavor gets sucked out right away and you're left with a unflavorful chunk of ice. And so we're removing that water. And as we remove that water, as it freezes, that means this concentration of solutes is now higher than it was before. And so since there's more solutes outside the cell, we have just basic osmosis and water leaves the cell. right? And so that cell then shrivels and dies because we've removed so much water. So the very simple solution that wood frogs have is to prevent this water loss is you add glucose into the cell. And this is what every freeze-tolerant animal does, not glucose necessarily, but others. And that prevents any further water loss. So now you have a nice stable environment. So that initial thing that these animals deal with when they freeze and why they make so much glucose isn't that they're scared of their cells being crushed by ice or their cells being burst by ice. It's simply dehydration and desiccation. They don't have as much water. Because it's frozen, they now need to prevent further water loss. And then once they've stabilized that environment, they can do everything else. And they can then worry about ice forming in the cell. When we get frostbite, which I am sure is a concern of so many people in California today, a little bit more of an issue for me, that's what happens is our cells actually dry out, dehydrate. So when we're watching this animal freeze, we're seeing this incredibly sugared up animal. That ice that we're seeing is external only, but because it has this ability to survive and produce all this sugar, it can then prevent that ice forming in its cell. And by doing that, it then can survive 7 months like this, down to -20, probably even colder. And come back to life and hop around. We probably won't be able to ever do this with humans, but we might be able to learn some things from these frogs when it comes to food preservation, as well as maybe organ preservation. And then, of course, this wood frog hops out and heads right to the water because it's going to get ready to breed. But I want to show you what that water looks like for these frogs too. This is from a couple of years ago, actually, and this is exactly what a wood frog pond looks like right now, a breeding pond. Here's me catching some frogs, and one thing you'll notice is there's ice in the background. So we call these animals ectotherms and cold-blooded. I want to point out these animals are mating, reproducing in ice-covered water. We think of the Arctic as a harsh environment. It's a harsh environment for us. This isn't the case for these animals. They're quite happy breeding at these temperatures. They're quite happy surviving a frozen winter. They're not as happy when it gets warm. I would say that when we call things cold-blooded, we sometimes use that as a misnomer because I don't think any of us would do very well swimming, let alone reproducing in this water. But these animals are quite well adept at this. And in fact, we can get them to call, and they'll be calling really soon. Here's what these calls sound like. And I don't want to play it for too long right now because I might get them calling me back tonight. So we have just an incredible animal that can do all these amazing things for such in conditions that we don't necessarily always think about. When you're thinking about the Arctic, yes, there's moose, yes, there's bears, but for all those big animals, we have so many other things underneath that soil or just hidden from plain sight that we can learn from and gain so much knowledge from on how we can survive winter or make things successful in our environments. So thank you so much for having me tonight. I saw there's some questions popping up, so I think I'll get those in a second.
Hi, Don. There are so many questions. So let's try and go through them. First one is, you mentioned, well, I guess, what triggers them to both start freezing and then also what triggers them to start thawing? Because you mentioned like touching ice, but yeah, what triggers them?
Yeah, it's only that ice formation on their skin. So you could freeze a frog in the summer. It's not going to do as well, but you could go up to a wood frog in the summer and drop some ice on it, and it will start this reaction if it's cold enough. So right now, things are starting to warm up because of that snowpack, right? That snow takes a while to melt. We have something called shoulder season here, and just in general where you have snow. So right now it's 50s and 60s. There's still snow behind me, in part because it's going to take a while for that snow to melt. So wood frogs are waiting for that snow to melt. Once that snow melts, they will then hop right out because the rest of the ground will thaw as well. And the same thing happens then in the fall too. It takes a while for the ground to freeze and for us to get that first snow. Once we get those things, then those wood frogs will also freeze. And they're just going to freeze as soon as ice touches them. So there's let me bring up a distribution map of wood frogs. So here's a distribution map of wood frogs. This one's a little inaccurate, but this is one of the best ones I have. They are all the way up here. Actually, we know they go into this area, but we have places that in the wood frog distribution that aren't going to freeze during the winter, or are only going to freeze for a few weeks at a time. So our Ohio counterparts are the more studied ones, but they can only survive about 2 weeks frozen. August, yes, it is a real background. I'm actually outside.
Kind of along those lines, there's also a question on like how can they complete this cycle multiple times? Like if there was a weekend snowstorm and there was ice and then there wasn't ice, do they
That's a great question, and that's something that we think there's going to be differences in our populations. With the northern frogs, are well adapted for producing a lot of glucose. And one of the things we know when they freeze is it takes a few weeks for them to actually freeze all the way. That is, it thaws at night and freezes. And so they have this ratcheting effect of slowly building up that glucose to those extremely high levels. That's why we see a difference in the lab and the field. But wood frogs up here, when they freeze, they want to produce as much glucose as possible and not resynthesize it back into glycogen. So they don't want to store it, right? They want to use it and then have it there. Versus these more southern species, what they're going to southern communities and populations, what they're going to want to do is they're going to have those multiple days of, I'm going to freeze a couple days in December, and then I'm going to be thawed, and then I'm going to freeze again in January, maybe February, a couple times. So those animals we think are instead going to be much better at resynthesizing, grabbing all that glucose they made and putting it back into their liver for their next freezing event. So we'll have like multiple ways to tackle the same thing. We'll have our lower southern populations that need to freeze multiple times for short periods. And then we'll have our northern populations that need to freeze just basically once for very long periods of time.
Yeah, and there was this one question on how does the wood frog get rid of the glucose so it just comes back into their liver?
So some of it does get reabsorbed and converted back to glycogen. Others gets urinated out. So they do lose some through the urine, but glucose is very expensive, especially for these animals that are about to breed. Breeding is also a very expensive thing. So they try and reclaim as much of that glucose as possible. And being at a lower temperature, that they won't get all the damage that we get from having high levels of glucose. They'll be able to reclaim a lot of that, and they'll be a little bit more tolerant to those conditions that would cause us to again die multiple times over from diabetes and comas.
That brings me to my next question: how do wood frogs not have diabetes?
And that's probably why we see them produce glucose only when they begin to freeze. It's both a very expensive molecule, right? You want all that sugar, but it also can cause damage if we have too much sugar available, glucose available. It interferes with proteins, it interferes with cell membranes, and different channels that we have in our cell membranes. So by doing it at a very low temperature, you're not going to have as much damage because there's not as much metabolic activity.
So if there is no cellular metabolic activity during their frozen state, do you think they might be able to survive frozen for multiple years or longer?
There is going to be some metabolic activity. immeasurably low. We can't measure this at that point. But I don't think they would survive years. One of the issues with freezing is recrystallization. That's what we see with our foods when we get freezer burn and things like that. We start to see those ice crystals reform in different ways, and that can cause that cellular damage that we're worried about. So we don't know how long they can survive. It's kind of difficult to create the right conditions in a lab to actually freeze them for long periods of time. It's definitely something we want to tackle eventually. Like I said, we don't know yet how cold they can go or for how long they can go. And we're still a ways from doing those experiments.
This might be another experiment, but what happens if a wood frog is moved to a warmer climate like Central California?
That is a great question. That's something that we are working on right now, is kind of this common garden thing of how do animals in the north in Fairbanks look if they live in lower latitudes where it's warmer, and how do those warmer climates do at higher latitudes. So, we're not going to see wood frogs in California, except in, well, they're not there, but let's say you introduce them, they're not going to be able to compete with the same species that do well in California because they're cold adapted, right? You wouldn't expect a moose to do well in California for the same reasons. And just like you wouldn't expect some very warm weathered animal to do well in Fairbanks. But we do have similar species to some of the species in California. In our lower parts of our state, in southeast and south central, we do have a few other amphibian species. Just the wood frog is up here in Fairbanks and in North of the Arctic Circle, though.
Cool. Well, thank you so much, Don. That was so interesting, and there's probably a lot of other questions if you want to take a look in the chat later. But thank you so much. I'm going to bring back Christina now to close out the night.
Thank you for having me.
Thanks. Hi.
Me pops. Sweet treat.
Thank you, everyone, for joining our Extreme Life night school tonight. Another special thanks to Jack, Valeria, and Don. We had a lot of fun, lots of information, very extreme. For those that weren't with us at the top of the show, we are back in person next week. We do still have night school. So in fitting with us returning to the building, next week we are going to visit the rainforest. So you'll get a behind the scenes from one of our own OSHA rainforest biologists. We'll also get to listen in on some animals of the rainforest and also hear more about Amazon deforestation and some paths forward.
And then it's my job to remind you to subscribe to our YouTube channel so you don't miss any future night schools. Yeah, and just for those of you in the Bay Area, we really hope to see you soon at Nightlife. And we'll also be showing night school at Nightlife in the building, so you can do both. But anyway, yeah, you can, I think we said we were going to drop a link, but yeah, calacademy.org/nightlife is where you can find out how to buy tickets and all that. But we'll be here. We love having you from all around the world, and we'll see you next week. Thanks again.
Night.